This is the lecture for Biology 104, Lab 13, Molecular Biology. Molecular biology is an area of biology that focuses on DNA and all of its processes. DNA is the molecule of information for the cell. It contains all the genetic information that determines what the cell will look like and what it can do. DNA is composed of nucleotides, and every nucleotide has a phosphate group, a sugar, and a nitrogenous base. And in DNA there are 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. The basic structure of DNA is a double-stranded helix. So this is an example of the twisted double-stranded helix, but every DNA molecule is made of 2 strands of nucleotides bound together. So this is 1 strand of nucleotides, this is 1 nucleotide. So the phosphate group, the sugar group, and the nitrogenous base. This is the second nucleotide that is covalently bonded to the first one. Again, the phosphate group, the sugar group, and the nitrogenous base. This is 1 strand of the DNA. The other strand of the DNA is over here, and again, these are examples of the nucleotides here. The important thing about DNA is complementary base pairing. Complementary base pairing means that adenine always lines up across from thymine, and guanine always lines up and pairs with cytosine. That's the complementary base pairing. So you have these 2 strands of nucleotides that are hydrogen bonded together with complementary base pairing, and then the whole thing is twisted. And that's the helix part. So it's a double-stranded helix. RNA is a little bit different. RNA has a different type of sugar, and also instead of thymine, it has uracil. DNA replication is an important process that involves DNA, and DNA replication occurs right before cell division. So replicating the DNA will make sure that each daughter cell will have a set of genetic information to determine what the cell will do. DNA replication occurs in the nucleus. And in this process, the DNA double helix needs to be unwound and unzipped by certain enzymes. And then each strand will serve as a template for replication. This process is called semi-conservative replication. So this is an example of DNA replication. Here is the original strand, so that's the double helix. There's a green strand and a red strand, so you can tell the difference. During DNA replication, this double helix will be unwound and unzipped. So the red strand will go over here, the green strand will go over here, and during the process of DNA replication, a newly synthesized strand will be made off of the template of the original. So here the original red strand is the template, and the yellow strand will be the newly synthesized strand. The green strand also serves as a template. So the green strand is from the original DNA molecule, and then the blue strand is newly synthesized. So semi-conservative means that in the daughter molecules of DNA, you have 1 original strand and 1 newly synthesized strand. This is another view of DNA replication with more detail showing the complementary base pairing. So this is the original DNA molecule that has been separated. The 2 strands have been unwound and unzipped to separate them. And this strand will act as a template, so an enzyme will come in. An enzyme will come in and start replicating DNA. So everywhere there is a cytosine on the original strand, the enzyme will create a new strand with a guanine. So cytosine, guanine, and everywhere there is an adenine, you will have a thymine added. The same thing will happen on the other side. So an enzyme will come in and perform complementary base pairing, matching cytosines to guanines, adenines to thymines. The second major process that involves DNA is considered the central dogma, or sometimes referred to as gene expression. And this is where the information that is encoded in the DNA is expressed in the form of a protein. So this is an example of the process. So information that is stored in DNA will be copied into a messenger RNA, and then that messenger RNA will be used to produce the protein product. The first process, going from DNA to mRNA, is called transcription. The second process, going from mRNA to protein, is called translation. The first step of gene expression is transcription, and transcription is where the DNA is copied into an mRNA. And this involves the information that is in the double-stranded DNA to be converted into a single-stranded RNA molecule. And this process occurs in the nucleus. And the adenine in DNA will be paired with uracil in RNA. So remember that RNA does not have thymine. It has uracil instead. This is an example of transcription. So here is the DNA double-stranded molecule, and when it's going to be transcribed, the DNA has to be unwound and unzipped so that the RNA polymerase, which is the enzyme, can come in and read the codons on the DNA and match the complementary codons for the RNA. And again, uracil is found in RNA only, so wherever there is an adenine, adenine is the green, wherever there is an adenine, it will be a uracil that will be paired with the adenine. So the RNA molecule will end up being single-stranded and will contain uracil wherever there would have been a thymine. Translation is the second process of gene expression, and this is where mRNA is used to produce the protein. This occurs in the cytoplasm of eukaryotic cells, and ribosomes are the main enzymatic complex that performs translation that forms the protein. And for translation to occur, you have to have codons, which are the 3 nucleotide sequences found in the mRNA. You need the genetic code, which correlates the codons to the actual amino acid. And then you need tRNAs, which will bring the appropriate amino acid to be put into the sequence to form the proteins. This is a table that shows you the genetic code. The genetic code is universal, which means all living things use exactly the same genetic code. And the genetic code correlates the codons. And remember, codons are the 3 nucleotides in the RNA. It correlates the codons with the appropriate amino acid. And most of the codons encode for different types of amino acids, but there are a few regulatory codons. The start codon is AUG, and that tells the ribosome where to start reading the codons and translating the RNA. And the AUG is important because this sets the reading frame. You also have 3 stop codons, so UAA, UAG, and UGA, and that tells the ribosome where to stop translating. So the ribosome is going to translate the codons that are found between the start codon and the stop codon. This is a different version of the genetic code, and it might be a little bit easier to read. And for this genetic code, you start in the middle and read the codon moving out away from the middle. And for this, for example, the start codon was AUG. So to find that out, you find the A, then you look at the U, and then you go to the G, and that shows you methionine, which is the amino acid that is included in the start codon. For the stop codons, they are the terminator codons here. So TER means stop. As an example of translation, here is a diagram which shows the mRNA. So the mRNA is the strip here, and it has the codons on it. So every 3 nucleotides equals 1 codon. Then this big blue complex is the ribosome. So the ribosome is sliding along the mRNA, translating the mRNA into a protein. Now the way this process works, you need to have the genetic code. So the very first codon the ribosome would have encountered would be AUG on the RNA. So remember, AUG is the start codon. So you look up the AUG, methionine is the amino acid in the start codon. Then that sets the reading frame. So after the AUG, every 3 nucleotides would be a codon. So for the process of translation, what the ribosome does is it reads the codon. So in this case, CGU. Then it matches the appropriate tRNA, and the tRNA has an anticodon, which is complementary to the codon. So if the codon on the RNA is CGU, then the anticodon would be GCA. So complementary base pairing, and remember, RNA has U, no thymine. So that tRNA would be attached to the appropriate amino acid. And to know which amino acid is attached to the tRNA, you need to look at the codon. So CGU, U, that's arginine. So this would have been an arginine. Then what the ribosome does, it shifts over to the next codon. It reads the next codon, ACG. It matches the tRNA that is complementary to ACG, which would be UGC. And then this amino acid correlates to the codon on the mRNA. So ACG, threonine. And the ribosome would keep shifting over to read the next codon, match the tRNA, and link the amino acid, and it will do that until it gets to a stop codon where it will stop. So as a review of gene expression, or the central dogma of biology, the first process is taking DNA and forming an mRNA, a copy of the DNA, into mRNA. That process is transcription. Then once you have the mRNA, you take that mRNA and produce the protein, and that is translation. DNA is also involved in a lot of different processes involved in biotechnology, one of which is DNA fingerprinting. DNA fingerprinting is used to identify individuals from each other, and it involves differences in the DNA sequences that different individuals have. And to perform DNA fingerprinting, a scientist would use enzymes to cut the DNA into different lengths, and the lengths depend on the sequences that the individual has. Then you would take those fragments of DNA, run them through electrophoresis, and compare the banding patterns. So an example of this, you have 2 individuals. This is individual 1, individual 2. You use the enzymes to cut the DNA, and because the 2 individuals have different sequences, they get different areas that are cut. So individual number 1 has 1 cut at that location. Individual number 2 has 2 cuts at 2 different locations. Then you take those samples of DNA, you run them on electrophoresis, which will separate out the fragments by size. So this separates the DNA, and then you compare the banding pattern of the two individuals. And every individual person will have a unique banding pattern. That's why they refer to it as DNA fingerprinting. Like every individual has unique fingerprints, every person has a unique DNA banding pattern. Probably the most common reference to DNA fingerprinting is in solving crimes. So in this case, you have a victim and you have DNA that was left at the crime scene, and you have three suspects, suspect 1, 2, and 3. So the DNA that was left at the crime scene, you first want to determine if it was from the victim. And as you look at these banding patterns, this pattern does not match the victim. So it does not match. So the implication is that this DNA came from one of the suspects. And as you compare the banding pattern, you can determine whose DNA that was. So if you look at this DNA, you have 2 bands up here, then 1, and then a bit of space, and then 3 that are spread out. Suspect number 1 does not have that banding pattern. Suspect number 2 seems to have that banding pattern, the same pattern. And then suspect number 3 has a different banding pattern. So from this, the DNA from the crime scene is identical to suspect number 2's DNA banding pattern. So suspect number 2 left their DNA at the crime scene. DNA fingerprinting can also be used to determine paternity, to find out who's the father. So every individual, like this child, their DNA came half from the mother and half from the father. So what that means is that every band you see in the child's banding pattern, in the child's profile, has to match either the mother or the father. So in this case, comparing the child to its mother, this band matches the mother, this band matches the mother, and this band matches the mother. So what that means is that the remaining bands, this band and these 2 bands, must have come from the father. So in order to identify the father, you want to find out which man could have donated that band to the child. So not dad number 1, so that rules him out, but it could be dad number 2 or dad number 3. Then you look for the next 2 bands, these bands that did not come from the mother. So you've ruled out dad number 1. Dad number 2 could have provided that second band, but not the first one, so it's not dad number 2, but dad number 3 could provide those last 2 bands. So dad number 3 is identified as the father. For your lab, you have 2 different DNA fingerprints that you need to answer questions for. And the first one is a crime scene DNA fingerprint. So it gives you a victim and 3 suspects and different samples of blood, and it shows you the DNA profile. Often when you're talking about profiles, you refer to different bands, and these are the numbers of the bands. So for instance, band number 1. Band number 1 is found in suspect A's blood, suspect C's blood, and suspect A's shoes. And then band 20, for instance, is found from the victim, the crime scene, and suspect A, and suspect A's shoes, and suspect C's shoes. So you refer to the band number. That's how you know which band you're talking about. The second DNA fingerprint is a paternity test. So this gives you a sample of the mother's DNA, the child's DNA, and 4 potential fathers. And again, when you are talking about different bands, to identify the bands, you give them numbers. So all of this is band number 1. All of these across here are band 10. Transformation would have been an experiment you would have done if we had been in class. And from the virtual lab you did before, you know that this is a way for bacteria to acquire new characteristics. And this occurs because genes from another organism are mixed with the bacteria. The bacteria will take in those genes, and then the bacteria produce the encoded protein. So the overall process You start with the bacteria, then you mix them with the plasmids, which is the DNA. And on these plasmids, there are two important genes. The first gene is for ampicillin resistance. Ampicillin is an antibiotic that kills most bacteria, but if they have this gene, they can produce a protein which makes them resistant to the ampicillin. The ampicillin will not kill them. The other gene is a gene that will convert galactose into a blue pigment. So if the bacteria are exposed to X-gal, then the bacteria will turn a blue color. So you would take the bacteria, mix them with the plasmid, then the bacteria would transform. So transformed bacteria are the ones who have taken in the plasmid and they can express these two genes. Then you would have taken the bacteria and put them on plates. And the plates would have different components in them. They could be plain luria broth, which is just nutrients. It allows all bacteria to grow. Some of the plates would have had ampicillin, and ampicillin kills bacteria that do not have the plasmid. Other types of media would have X-gal, and so the transformed bacteria, the bacteria with the gene, with the plasmid, would have been able to convert the X-gal into a blue color, and those colonies would have turned blue. So with the information from the virtual lab from last time and the information that is written in your lab report, you should be able to predict if the bacteria will grow on the different types of media and what the color will be. The normal color of the E. coli is a white colony, but if they are able to produce the blue pigment, then they will turn blue.